Your career begins here at Satyabama Institute of Science and Technology, a deemed to be university, a nationally recognized and globally acclaimed destination to accomplish your academic aspirations. The Colonel Dr. Satyabama Institute of Science and Technology, with the International Research Center, houses advanced research facilities that have 13 centers of excellence, 10 plus technology transfers, 100 plus patents, 10,000 plus reputed publications. The Satyabama Center for Advanced Studies is a state-of-the-art facility built with the aim to cater to the futuristic aspirations of our students with the aid of a metaverse studio, drone labs, maker's space, media center, advanced characterization facilities, ICT-enabled classrooms, and supercomputing laboratories. The engineering departments are equipped with sophisticated infrastructure, fulfilling the academic and hands-on requirements of our students. The architecture department is furnished with sophisticated design studios and spacious drawing halls, all of which support the learning process of our postmodern generation architects. The Satyabama General Hospital, along with the School of Dental, Nursing, Pharmacy, and Physiotherapy, have advanced clinical facilities. The monumental Central Library houses a diverse collection of books, periodicals, and journals. Additionally, it also has an open-air theatre with a seating capacity of 1,000. The Management, Arts, Science, Law, and Hotel Management departments comprise of all the necessary and required facilities, creating the perfect ambience for our far-sighted students and catering to their skill set needs with the end goal of equipping them to live their dream. Our chain of laboratories with practical exposure equips our students to build a successful career. Our institution has a team of dynamic and outstanding teachers who are ever reachable to our enthusiastic students, promoting the spaces to learn and places to grow. Greenery around the campus greets and accommodates our students, who are the backbone of a developed nation, edging them on to achieve the summits of their success. Satyabama Institute of Science and Technology.
Good afternoon, everyone. Welcome to the Ocean Research Knowledge Series, Day 6 afternoon session, organized by Center for Ocean Research and National Facility for Coastal and Marine Research, Satyabama Institute of Science and Technology, aimed at promoting knowledge and innovation in ocean sciences. Today, we have with us Mr. Raghavendra Kumar to give a lecture on the topic Fish Diversity Assessment Using Environmental DNA Techniques. We are happy to have you with us. Over to you, sir. Thank you.
Thank you. Am I audible?
Yes, sir. Yes, sir. You are.
Thank you.
Please share the screen, sir.
Yes. Shall I start this?
Yes, sir. You can start.
Yes, thank you. Good afternoon, everyone. I'm Raghavendra Kumar, Assistant Professor, National Facility for Coastal and Marine Research, Center for Ocean Research, Satyabama Institute of Science and Technology. And my core area is genomics, and I am here to deliver a talk on fish diversity assessment using environmental DNA techniques. So before going to the session, I would like to give a short introduction about the facilities and services available in NGS Hub and Advanced Biovisualization Lab at NFCMR. Yeah. So this is the facility where which supports the work which I'm going to talk about. So we have Illumina Nestic 2000, Omega Spectrum Compact System, Asanga sequencer, and digital PCR, and then many an MK1D nanopore sequencer. And we do have a high-end workstation and the server which supports bioinformatics and others. So what are the services we provide? We provide whole genome sequencing, transcriptome sequencing, eDNA metabaptism, shotgun metagenome sequencing, and Sanger sequencing. And apart from that, in bioinformatics lab, we have a Schrodinger computational platform with which we can provide a drug discovery analysis. And apart from that, we do have an open source omic data analysis. And then primary objective of this lab is to integrate marine genomics for monitoring marine biodiversity, ecosystem health assessment, evaluating the impact of pollution on marine ecosystem, and effect of climate change. We do have a collaboration with scientists, researchers, industrialists, and experts. So marine genomics is a fast-growing area with more research opportunities and job opportunities. And yes, this marine genomics is part of our MSc Marine Biology course. And with this, students can benefit or they can get a practical exposure in modern genomic techniques. So let me start with my session. What is environmental DNA? So before talking about environmental DNA, I want to give a small example. We have to we are collecting some soil samples and we want to identify what are the bacteria present in that particular soil sample. So normally what we do is we do a serial dilution, inoculate or culture, and then using biochemical or morphological characteristics, we identify the particular bacteria or bacterial community present in that particular soil environment. So in eDNA, using eDNA, what we can do is just isolate the DNA from the soil, do a NGS library preparation and sequencing followed by bioinformatics analysis, by which we can identify what are the bacteria present in the particular soil environment. Same thing we can apply for fish diversity study. Here we have to collect a water sample do a DNA isolation, and then followed by NGS library preparation, again sequencing, then analyze. Using bioinformatics, we can identify what are the fishes present in the particular water environment. So, yeah, what is environmental DNA? Environmental DNA is a genetic material fed by organisms into their surroundings through stem cells, mucus, gametes, fecus, or decomposing tissue. So, what are the types of, what are the sources of environmental DNA? Water, soil and sediment, air, ice, and permafrost. So, why it benefits? Like eDNA is a non-invasive, no capture, no harm to organism, detect rare, cryptic, or low-density species, scale to whole community biodiversity survey, and applicable from macroorganism to micro. So this is evolution of eDNA studies. It all started in 1987, where a group of scientists optimized the first eDNA extraction protocol. And in 1980, the first DNA metabarcoding study started. And in year 2008, the fish species was identified. Fish species present in freshwater samples were identified using this eDNA technique. Then 2010 is an NGS era. eDNA has expanded rapidly, and we were able to do complete. We were able to identify the complete community present in particular from a single sample or present in particular environment. So there are two forms of eDNA: intracellular and extracellular DNA. So intracellular, the name itself is telling intracellular DNA present inside the organism, bacteria, archaea, and microeukaryotes. So extracellular DNA is a DNA outside the cell, which is free DNA. It can be called as free DNA, and it is mostly released in the environment, and it is free floating in the water or settled down in the soil. So this eDNA can be done by two different approaches. Yeah, two different types we can do. One is metabarcoding, and another one is shotgun metagenomics. So metabarcoding, we have to amplify a specific marker gene to identify multiple species within a target group. Like specific marker genes means each organism have its own specific marker genes. For example, bacteria has 16x ribosomal RNA, fungi has ITS, and fish has COI. So here, a favor for rapid biodiversity assessment, detect up to 3x more species than traditional survey method. Best suited to target group specific survey. And shotgun metagenomic sequence, here we have to take the complete DNA directly to the library preparation, then followed by sequencing and bioinformatics analysis. In data analysis, we can use a specific database belongs to specific organism. So, what is the sampling media and genetic markers? Yes, for fish, we can use cellulose ribosomal RNA or COI, and the typical medium will be water. And bacteria, the genetic marker is 16x ribosomal RNA, sediment, or rhizosphere, or we can use water also. So fungi, it is ITS and sediment. Microbial eukaryote, 18S ribosomal RNA, sediment or water, and bird mixed metabarcoding. Yeah. So yeah, this is the complete workflow for eDNA analysis. It all starts with sampling. So we can collect water or air or soil or sediment. So the like we should have some care how during the sample collection. Like key points are non-invasive sampling, collect, replicate, then prevent contamination and store sample appropriately in a proper storage condition. Actually. Okay. Then once the sampling is done, it should be immediately processed for the DNA extraction. So it should be taken within a 24 hours to the lab, and we have to start doing the DNA instruction. So DNA instruction, the key points are it should have a efficient cell life. It can be a conventional T-tab method or a kit-based method. Each sample we have to optimize our own DNA isolation technique. We have to do more validation and we have to identify yes, this is the DNA method we can use for our specific sample. Then it should be this isolation method should be careful in removing the inhibitor. And we have to assure that we are getting high purity environmental DNA. This can be quality check using qubit nanodrop and gel electrophoresis. So DNA instruction is the crucial step. We have to take more care on doing the DNA instruction. Then this is followed by after DNA instruction, we have to do a library preparation. Library preparation, as I told earlier, we can use two different methods, metabarcoding or shotgun metagenomic library. So metabarcoding is nothing but we have to take a specific markers, we have to amplify that, adding adapters, then proceed for the sequences. In metagenomic library preparation, we have to take a complete DNA directly to the library preparation, then followed by induction and then sequencing. These are actually two different methods. In metabarcoding, we can use specific marker as I told earlier. Like for fish, we can use COIG. For bacteria, we can use 16 ribosomal RNA. For fungi, we can use IPS. And once the library is ready, we have to do a quality check. This quality check can be done in like we have to do it two different steps are there. One is concentration. Concentration can be done using qubit and another one library fragment size. This can be done by tape sequence. So sequencing can be done using Illumina, Nanopore or other NGS platforms. So once the sequencing completed, we have to go for bioinformatics analysis. And bioinformatics analysis, first initially we have to do a quality control of raw data, part-species and multi-species. Using this tool, we can do a QC check of raw data. So this is followed by quality filtering and trimming. This is trimmomatic. A tool called trimmomatic. We can use this for quality filtering and trimming. Then this is followed by sequence filtering, clustering, ASV. ASV is amplified sequence variant. The tool uses data2 and followed by taxonomic assignment. In this taxonomic assignment, we have to use different database. For bacteria, we have a similar database, and GTDB is there, and then NCBI is there. And for fish, we use Midori2. And then, so these are the applications like population dynamics, ecosystem health monitoring, biodiversity assessment and conservation and management. So this is the studies, some of the studies in DNA techniques, and the first one is it happens in 1987. It has been done in 1987. Here this is the first eDNA instruction. This is like eDNA optimization has been done in this study. So this is the first study to do the eDNA optimization, extraction optimization. So they have taken two sediment samples are collected from a marine slug and another from brackish water near Pensacola, Florida, and the third one from a reservoir near Knoxville, Tennessee. So they have optimized with the normal conventional method using their own lab chemicals. Now people have started using different kit-based methods. So study 2 and study 3 is there, and let's start with study 3. This study focuses on invasive marine species using environmental DNA collected from water and sediment samples. The researchers successfully identified both native species and newly introduced invasive species without physically collecting the organism. Early detection of invasive species is important because they can spread rapidly and disturb marine ecosystems. Yes. So next is study 2. Here the researchers at the University of Washington showed that airborne environmental DNA can be used to detect and monitor the migration. Yes. Can be used to detect and monitor migrating salmon population by collecting air samples near a stream. They successfully identify salmon DNA without sampling water. So here they have used a gas sample to identify the salmon fish population, whereas study 3 they have used a water sample. In Great Lake and the study 4 is Great Lake USA, early detection of invasive Asian carp. So carp, silver carp retain native fish population in the Great Lakes. Traditional survey often failed to detect fish at low abundance. So water samples were collected from rivers and canals connected to a Great Lake. And the species-specific PCR detected Asian carp DNA before fish were physically captured. eDNA provided an early warning of invasion. Authorities used the results to strengthen monitoring and management. So here the study 5 is detecting rare and endangered species. And for that, the researchers collected water samples from a stream and eDNA was detected from the species that was missed by normal netting, normal fish netting. Detection probability was significantly higher compared to the traditional fishing survey. And this is our finding. So here, this is our first eDNA study in our lab. So in this study, we have collected water from Sadambakam Distillery. 5 liters of water we have collected, and this 5 liters of waters were allowed for filtration using vacuum pump, and we use a 0.45 filter. So once the filtration is done, this filter which captures the DNA was dipped into a lysis buffer, that is CTAB lysis buffer, and this is followed by the DNA isolation. Once the DNA isolation is completed, we have done the usual quality check using agrogellar electrophoresis and filter. Once it is done, we have used this study is for identifying fish diversity in the particular estuary. So we have used the COI gene. That COI gene was amplified. Using this DNA, what is the isolated DNA, we have amplified the COI gene. This is now, after amplification, we have used the library preparation kit by which we added an index and like adapter, and then this is followed by the sequencer. That we have done the sequencing in nanocode sequencer, and then it is followed by the bioinformatics analysis, and we use the Midori 2 database. The Midori 2 database is the reference database for eukaryotic mitochondrial DNA sequence. So what we identified is we got some of the freshwater fish and shallow coastal water fish and saltwater fish. Because this, like, these estuaries is mixture of freshwater and marine water. So there is a possibility of getting more freshwater fish and also the marine water fish. So monitoring beyond fish and microbes, yeah, of course, we can do a bird DNA analysis. This is helpful in migration survey, then megafauna, foraminifera, and IUC treated taxa. Then limitation and technical challenge. So reference database gap. Yeah, for some of the organisms, there is no specific genomes available in the database. So for our organism, we may, when we are doing those analysis, we may end up in end up at the level of family or gene. So this need to be, we have to, all over the globally, it need to be, the organism, more organisms need to be sequenced. So sediment extraction hurdle. Yes, sediment sample has humic acid and tannins which can affect the DNA extraction and also it inhibit the PCR. So environmental sensitivity. Different environments have different temperature, sunlight, and water flows are different. So it can affect the environmental DNA, eDNA present in the particular environment. So we have to take like season wise it should be collected. Different season we have to collect and do the eDNA. So site specific protocols needed. Yes, sir. For each site we need to optimize different protocols. So it depends on the sample types also, water or sediment sample. We have to do optimization for DNA isolation. We have to do optimization for library preparation, so sequencing, everything we have to do for the system. Then validation and standardization. So before going to eDNA, before doing eDNA analysis, we have to, we should every lab should have a in-house reference data. Like we have to collect the samples, do a Sanger sequencing, and like in case of fish, we have to collect the fish samples. We need to do a COI sequencing, and this we can collect as our own database. So uh, starting with in-house reference database is the best way to start the eDNA technique. And this can be, we can get the help from a traditional technique for this process, which will increase the power of your eDNA technique. So here's a direction and resource gap. Restoration framework. Yeah, eDNA can be used in restoration ecosystem and climate resilience. We can study the effect of climate change using eDNA techniques and standardized workflow. Yes, there is no standardized workflow for globally. So that means there is no proper guideline. So there is a requirement for a guideline for DNA extraction, for library preparation, and for the markers which we have to use and the analysis. For each and every step, we need a guideline which is globally accepted. And then expanded task. Okay. Here we can increase, we can expand the taxon to like other organism also we can do with eDNA techniques like birds, insects, or fungi. So paradigm shift for coastal conservation. Environmental DNA provide a rapid, invasive, and high sensitive means of assessing biodiversity. And yeah, eDNA is a standard tool in the globally. It is becoming a
Excuse
standard
me,
tool,
sir.
which is useful for conserving and restoring ecosystem. This eDNA technique can outperform traditional survey for cryptic and rare species detection.
Sir Raghavendra, sir, can you hear me? Sir, actually, your screen is not fully visible, sir. It is some other tab is also aligned and open, sir.
Okay. Okay.
Yes, sir. Now it is fine, sir.
Yeah,
Thank
I'm going to conclude this. Outperform traditional survey for cryptic and rare species detection. Extend from fish to fungi. It can be extended to fish to fungi, bacteria, birds, and treated other treated factors. Ready for integration in restoration. So yeah, genomics is language of all life. Decoding the stories written in every organism from microbes to mammals. In the near future, it will become the lens through which we understand the ecosystem evolution and every living cell on Earth. Thank you.
Thank you, sir. Thank you for your wonderful presentation, sir. So participants, if you have any questions regarding the topic, you can post it in the chat box. Okay. There is no questions in the chat box, sir. Can we conclude the session?
Yep.
Yes, sir. Thank you, sir.
Thank
So
you.
if you are interested in oceans, marine life and research, enroll in our 2 years MSc Marine Biology program and gain hands-on experience, research training and valuable career opportunities. So begin your journey by participating in Ocean Quest 2026, our free online marine science quiz. Register today and receive an e-certificate. For updates on upcoming programs, workshops and events, kindly visit the Satyabama website and follow the Center for Ocean Research social media pages for the latest announcements. The brochure and registration details are available in our chat box. So don't miss this opportunity to begin your journey in marine sciences. Share this information with your friends so we can join tomorrow by 11 a.m. Thank you.